microbiology, which is an extremely prestigious award. Research in the Nodwell lab that we're going to hear about today focuses on an area that's near and dear to my heart, which is bacterial communication and the use of small molecules for various purposes, like it allows bacteria to communicate and coordinate their behavior as a unified group and can allow them to acquire nutrients that are in small quantities. And they can also be used as weaponry to knock out the competition. So his research really straddles fundamental biology and also applied biology, and that these small molecules that have toxic effects may have potential as antimicrobials for fighting infectious diseases. So with that, if you'll join me in welcoming Dr. Nodwell.
Thank you very much. Thank you very much, Terry. I've had a terrific visit. I had great meetings with everybody, and I really enjoyed lunch with the students as well. That was terrific to get to know all of you people. I guess how to describe the work in my lab. We are what I like to think of as a chemical biology lab, which many people like to point out is just biochemistry reversed, but it does actually have some significance. The way that work in my lab usually proceeds is we start with a biologically active small molecule, usually like a drug or a chemical probe of some kind that has an effect on cells that we're interested in. And then we use that molecule to try and discover how it works. What does it interact with in the cell, and what happens to the cell when it does so. So in that sense, what we study in terms of cell life is driven by the molecules that we discover through various kinds of small molecule screens that we do and other foraging expeditions that we go on. The lab's about 12 people, and we have projects dealing with fungal biology. We have a project dealing with pathogens. And the work I'm going to tell you about today concerns a genus of bacteria called Streptomyces. And these are fantastic bacteria to work with. They're technically challenging, but they're one of the first things that drew me to them and many other people, I think, is that they're just beautiful looking. They have many visual cues. So, for example, this is Streptomyces silicolar, and you can see that the colonies are secreting a molecule, a blue molecule, into the medium. That molecule is called Actin. It's an antibiotic. I'll have more to say about that later. The colonies are also covered with a layer of white fuzz. That's significant, and I'll talk about that as well. And so the point is that these bacteria, they have many different phenotypic cues that you can mess around with, and when they change, you can use that change to learn something about their biology. And oftentimes the biology of Streptomyces informs the biology of all bacteria in ways that is surprising and interesting. So the thing about these bacteria that is most famous is that they're the source of many of our drugs. They're the source of almost all of the antibiotics that we use clinically. So you're looking at 4 examples here. Tetracycline and erythromycin are quite famous antibiotics. Some of you in this room have almost certainly been on both of these, one or the other, or possibly both of these drugs at one point or another. And on the bottom there's an anti-cancer drug, daunorubicin, or derivatives of daunorubicin are used for cancer therapy, and rapamycin, which is used as an immune suppressant. Now, so chemists and biologists argue, I think quite fruitlessly sometimes, about what the best source of drug leads is. Is it from synthetic molecules, which are easy to make and easy to modify, or is it from these kind of natural products? And I think that's a very silly argument. There are good drugs from both sources, and so really there's no disagreement. The strength, however, of the natural products is that they are ancient molecules. They've existed for hundreds of millions of years. They're products of evolution, and they're made by enzymes. And the thing about enzymatic synthesis is that there's a control of chirality. So if you look at these molecules, for example, if you take erythromycin, you can see that there are, if you count, there's 14 chiral centers in that molecule. The bacterium that makes erythromycin makes a single enantiomer of it. For a chemist, that's a huge challenge. And so the advantage of these molecules is that evolution has done the work of driving them to bind very specifically to their targets. So that's one of the reasons why people are interested in them. So they've been quite useful as drugs. This is a list of some of the antibiotics here, and you'll see many familiar names there. And you can see that they're almost all produced by species of Streptomyces. There's a few close relatives, Saccharopolyspora and Amyclotopsis, which are very, very similar genus to the Streptomyces. There's also the immune suppressants, the chemotherapeutic drugs, and antifungal drugs. So they really are a source of much of our pharmaceutical matter, and that's one of the things that's been most important about them over the years. They're also just really interesting and unusual bacteria. Almost everything you look about them is different than what you're used to when you think about a bacterium. So first of all, this is a schematic map of the Streptomyces celicola chromosome. It's about an 8.3 million base pair molecule, so it's quite large. It's a linear molecule. It has telomeres, so it has many of the properties of eukaryotic chromosomes, except that it's a true bacterium. And then it has other things that are color-coded that we don't need to talk about. But then one of the really remarkable features is that Streptomyces celicola, and in fact all Streptomyces, make anywhere between 25 and 50 different what are called secondary metabolites or specialized metabolites. And these are these biologically active small molecules that have been so successfully exploited as drugs. So this slide's a little out of date, but the principle holds for most Streptomyces. So one thing you find is if you compare genomes of different Streptomyces, there's a small number of the secondary metabolites that they have in common. So for example, desferrioxamine is a very common siderophore. Isorenaritine is a molecule, we don't really know what it's for. It absorbs light. It's the only other genus of bacteria that uses it is a species of green sulfur bacteria, and they use it in a sort of a ancestral photosynthesis-like reaction. My gut is that what it does in Streptomyces is that it serves as basically suntan lotion. It's a photoprotectant, but no one's studied that. There's a signaling molecule there, so a quorum-sensing molecule, SCP1. And then there's all these other compounds. So in terms of antibiotics, we have a calcium-dependent antibiotic at the bottom, the circular one. That's a peptide antibiotic. It's similar to daptomycin, which is used clinically. There's actin, which is the blue molecule up there on the top. There's prodigonine, and then there's many others. And each of the black lines indicates the location of the genes that make each molecule. And one of the really convenient things, possibly one of the only convenient things about this field, is that the genes that make the biochemical pathway that synthesizes each of these molecules tends to be clustered. So if you find the cluster, you find everything that the cell needs to make that molecule. And in fact, you can move the cluster from one species to another and drive species to make molecules that they don't normally make. So I will come back to that in a while. Okay, so in addition to its remarkable chemistry, Streptomyces also has a bona fide developmental system. It has a developmentally programmed life cycle that has a clear beginning, middle, and end. So the beginning of the life cycle is spore germination. The spores are just little oval cells, and when they land on a suitable growth medium, I should say the Streptomyces are normally found in the soil. You can go to any soil anywhere on Earth at the bottom of the ocean or just outside this building, and you will find many, many, many different Streptomyces growing there. The diversity is quite astounding. We have no idea what the total number of species is. But anyway, they make spores, and when the spores germinate, they grow cells that are filamentous rather than the usual sort of rod-shaped or coccoid bacteria that you're used to from what most people think of as bacteria. So the first cell type that you see, they're called substrate hyphae, and they grow by elongating. They grow from at the tip. All the cell wall biosynthesis is at the tip. So they extend the tip, they replicate their chromosomes, and they distribute them across this filamentous cell, and the cells can branch as well, so that eventually what you get is a mycelium that looks a lot like a fungal mat. You see this dense network of filamentous branched cells. To the naked eye, it just looks like it could be E. coli. It's just a beige blob on a plate. But if you let it go for a day and a half or 2 days, then something quite remarkable happens. You get a layer of white fuzz on the colony surface. And this is a completely differentiated cell type. And these are called the aerial hyphae. And they grow up into the air, And so you look at your colonies one day, and they are just beige blobs. You come back the next day, and they're covered with this fine layer of white fuzz. The aerial hyphae are the site of spore formation. And so what happens it's really just an example of developmentally regulated cell division. It's worthwhile to pause for a second and say that the genes and the proteins that drive septation, the sporulation septation event in Streptomyces, are almost identical to the genes for cell division in Bacillus subtilis, E. coli. There's a protein called FDZ that marks the point where cell division is going to happen. You assemble a divisome, and then you divide at that site. And it's all true for Streptomyces, except that it's as if these really fundamental processes, they're conserved, but they're deployed differently in space. So the substrate hyphae down here, these guys, don't really divide. They just get longer and longer and longer. Occasionally they lay down a cross wall that gives them a certain amount of structural integrity, but they don't undergo true cell division at all. So they don't do cytokinesis, they make cross walls, but that's it. They consist of long compartments of maybe 20, 30 chromosomes each. The aerial hyphae go through this developmentally regulated and concerted round of cell division that divides them into little compartments. And then there's a developmentally regulated chromosome segregation event that puts one chromosome into each compartment. And then there's a morphogenesis event, and these compartments mature into the spores. And one of the really convenient things is you have there's a sort of visual cue for the completion of spore formation, because most Streptomycetes will make a pigment and deposit it in the wall of their spores as the very last step in this whole life cycle. So one of the species I'm going to talk about later, Streptomyces venezuelae, at the very end of its life cycle, the spores turn green. And so the surface of the colony goes from white to green. And it all happens in a very orderly fashion. So this is a slide I love to show. So this is a cross-section of a colony of Streptomyces silicola just growing on agar. And if you look at it, you can see, where's my little thing here? This is the white layer of fuzz here. So these are the aerial hyphae growing up into the air. So that's where the spore formation is going to happen. Then this is all the substrate mycelium down below the agar surface. And you can see that there is one region here where there's a red pigment being made. That's a molecule called undecylprotegiosine, which is a secondary metabolite that is active against eukaryotic cells. We know very little about it, actually. And then you can't see the color so much, but you can see this region here where there's another pigment, and maybe you can see that that is blue. That's the region where actin is being made. So you have extraordinary division of labor within this colony. You have different cells making the blue pigmented antibiotic and the red pigmented antibiotic, and there are another 25 secondary metabolites that we can't see in this slide. So fascinating division of labor. It really is like a little organized community with heavy division of labor between one cell type and another. Okay. So I told you that we're a chemical biology lab. I'm going to tell you about two stories that involve the use of small molecules, and these are synthetic molecules, to perturb this life cycle in various ways. Okay? So basically the idea is you add a molecule to cells and you try to see something change. So we're starting with chemical perturbation of secondary metabolism. So this was a really remarkable project that was carried out by a graduate student named Erin Craney. She just took a faculty position at Cornell Medical School in New York. It's a really bright light, and it's a remarkable piece of work. So let's return to this for a second. You can see all the black ticks, which are the sites of the gene clusters for the molecules we know about. Those are molecules that can be purified from a normal wild-type colony. You can also see a number of white ticks along the chromosome. And those are also gene clusters for secondary metabolites. And this is the case with all of the Streptomycetes. We don't know what most of the molecular products of those gene clusters are, and we don't know what the biological activity of those gene clusters might be. And one of the interesting things is if you go and if you look at the genomes of different Streptomycetes, like I say, there's a few common compounds encoded in each of them, but then there's extraordinary diversity. No two Streptomycete species have exactly the same set of secondary metabolites. The result is we have no idea what the upper limit is of these molecules. There are presumably tens of thousands of distinct secondary metabolites. And so the problem is that these these so-called cryptic secondary metabolites tend not to express well in the lab. So you can take the bacterium out of the dirt, grow it on a petri dish, that part's easy, but most of these little treasures are not expressed, and so you can't purify them, you can't study them, and we don't know how they work. So Aaron Craney's project, which really kicked this whole story off, started with the question, can we use chemical perturbation to turn on some of these cryptic secondary metabolites? The reason for doing this is obvious. There's a very significant need for new antibiotics. We have rising antibiotic resistance everywhere. There are clinical pathogens that are resistant to all of the antibiotics that we have at our disposal. And so there's many efforts around the world to try and find new antibiotics. And so ours was aimed at trying to mine the cryptic secondary metabolites for new compounds. So Aaron took advantage of this blue pigmented compound, actin. And what she did was she set up culture conditions that she could A, do at a 96-well plate, and B, where actin was not synthesized particularly robustly. It's just a matter of manipulating the medium. So if you look along the top row there, you can see she made a gigantic spore prep of Streptomyces cilicolor. She plated 15 spores in each well. And so you can see that along the top row, you can see that there's a red pigment being made, but not very much of the blue pigment, actin. The bottom, the 8 pairs of rows of wells on the bottom are duplicates of molecules 1 through 8, sort of chosen at random. And you can see the compound number 1 has reproducibly triggered the production of the blue pigmented compound. So this sort of visual cue was what Aaron did to try and find biologically active molecules that perturb Streptomyces cilicolor. And I have to say it was one of the most fun things we've ever done in the lab. She screened 30,000 compounds by hand in duplicate. It took about 6 months, and once a week she would walk into my office and just say, look at this, and she'd hand me a 96-well plate with some freaked out thing happening in a well due to a synthetic compound doing something weird to Streptomyces cilicolor. It was really a very exciting project. She collected a group of we have about 10 that we've been working on since, and these are compounds that reproducibly trigger the production of actin. And so the structures are shown here. She named them ARC, which stands for antibiotic remodeling compound, although she confessed after her paper was accepted that it actually stands for Aaron Rachel Craney. So one way to achieve immortality, I guess. And so all these molecules, I'm not going to tell you about all of them. I'll tell you that they all do different things. They all act on diverse pathways, and most of them work on any streptomycete you try them with. You add them to a streptomycete species, you will activate some of the cryptic secondary metabolites. So I'm going to focus on this guy for the rest of the first part of this talk. And just a little bit more background on this. So the questions are, to me, the exciting thing is how do they work? What are they doing to cells that cause the cells to activate these molecules? And can we use this as a way to find cryptic secondary metabolites that might be interesting for drug discovery? So let me show you a little bit of actual data here. So this is an LCMS trace of an environmental isolate of Streptomyces, chosen more or less at random, and incubated in the presence and the absence of this compound ARC2. So that guy, the red circled guy. And so the blue trace is from the culture. So what we did was we grew the cells in the presence or absence of ARC2. We made a small molecule extract, and then we analyzed those extracts using LCMS. And the blue trace shows the untreated culture. The red trace shows the treated culture. So you can see that there's one peak that is unchanged. That is a well-known secondary metabolite called actinomycin D. So it's been studied for years. We know how it works. We were not that interested in it. But if you look at this guy, he's kind of a humble little peak in the untreated culture, and it's now the dominant peak in the ARC2 treated culture. So one of the things that we've been doing is just purifying molecules that get upregulated in this way, assuming that they might be cryptics. And we found quite a large number of molecules. We've defined a thing called the ARC2 elicitome, if you will, the collection of molecules, some of which we see over and over again, that are activated in the presence of ARC2. 2 molecules that we're particularly interested in are shown here. These molecules have fairly potent antifungal activity. The one on the top, we know how it works. It's an inhibitor of an enzyme called the V1 ATPase, which is involved in vesicle-mediated trafficking in eukaryotic cells. The one on the bottom is called 9-methylstreptomidone, and we don't know how it works, although I'm quite interested in it. Someone was asking about postdoctoral positions earlier. It's a rip-snorting project, let me tell you. Let me see if I can find. So this is basically, this is yeast growing here. This is a filter disc with, in this case, oxohygrolidin. In this case, a couple micrograms of 9-methylstreptomidone. And what you're looking at here is a zone of inhibition where the yeast has failed to grow in the presence of, because of the presence of the drug. So you can see there's a very big zone of inhibition as a result of 9-methylstreptomidone. It's really a very potent antifungal. So we're quite excited about this, and we're interested in figuring out how it works and whether or not it has potential clinical use. So that's some of the sort of applied stuff that we've done with these chemical elicitors. To go back to the sort of more basic science questions, which is, we don't really understand how secondary metabolism or specialized metabolism, whichever you prefer, how it's regulated, how it works. And so we did an experiment, and this has taken a long time, but we did sort of the same thing. We grew a culture in the presence and absence of ARC2, except instead of looking at the secondary metabolites that are being produced, we harvested RNA from the two and used RNA-seq to find out which genes are being turned on preferentially in response to this compound. And so this is a busy slide, and it's kind of hard to see. The lines are a little fine. But so the line is a representation of the Streptomyces celicolar chromosome, and the genes ordered along the length of it as they exist there. And then the lines going up and down indicate the genes that are activated or repressed by the action of ARC2. And you can see that there's a very large number of gene changes that happen. It was gratifying to see that quite a few of these genes are genes for secondary metabolites. And so there you can see that the biggest response is actin and the calcium-dependent antibiotic, prodigenine, and many others. Excitingly, there was also the antibiotic there was a Cryptic, and the hopine was also an unknown compound at the time. So this is basically what this thing does. It really does remodel antibiotic production. There's also a number of secondary metabolites that are downregulated. So isorenaritine goes down for some reason, SAP-B goes down, the iron siderophore goes down, and melanin and a deoxy sugar, and so on. So it's a very dramatic effect that this has. One of the other things we found was that there was a number of signal transduction genes that were upregulated and downregulated, and one of them really stood out. And that is a pair of genes called AFSR and AFSS. So these genes were first discovered by a guy named Suehara Horanouchi in the 1980s. And he discovered them in a screen where he made an overexpression library, so he could just randomly clone genes from Streptomyces celicolar, transformed them, and looked for genes that upregulated actin when you turned them on. And one of the things that he found was AFSR and AFSS. There's been a little bit of work since then on them, but not a great deal. And the model that existed a couple of years ago was that AFSR was thought to be a phosphoprotein that would be phosphorylated by a protein called AFSK, which is a membrane-associated serine threonine kinase. Once it's phosphorylated, AFSR can turn on AFSS. AFSS is a protein of unknown function. We don't know what it is, but we know that it does have the capacity to activate secondary metabolism, or at least there's one or two papers that support this. And so AFSS is thought to be the gene that actually turns on the secondary metabolic genes. So, and this was the only, of all the genes that went up and down, this was the only one that we had any sort of clue might be directly related to secondary metabolism, aside from the secondary metabolites themselves. So, another student in the lab, Vanessa Yoon, created a fusion of the AFSS promoter to a Lux gene reporter that makes luminescence, basically, when the genes are turned on inside cells. These are based on the photorhabdous luminescence cluster. We rebuilt them for Streptomyces, if there's anyone interested. And then she asked the experiment, could she turn on her reporter with ARC2? And the data was pretty convincing. What you're looking at here is a time course in the presence, so the blue line is just she added solvent, which is the DMSO, and then increasing concentrations from 10 micromolar to 50 micromolar of ARC2. And you can see there's a pretty clear dose response that activates the AFSS promoter. So we thought we're clearly onto something. So the next thing she did was to create a mutation in AFSS, just knock the gene out in the chromosome, and then look to see what effect that mutation had on the action of ARC2. And the result, again, was quite clear. So if you focus on the left, you're looking at there's a control culture that has been treated. These cells have been growing for about 2 days, so they haven't started making any actin erodin yet. The culture on the left has had a little bit of the solvent DMSO added, and then there's duplicates which have had ARC2 added to them. And you can see there's been a very strong, robust production of actin erodin induced in response. If she knocks out AFSS, this effect is almost completely obliterated. There's no response at all to ARC2. So AFSS is clearly important for the ARC2 response. She next knocked out AFSR, and I'm not showing you the data, but she had exactly the same result. If you knock out AFSR, you also don't get any actin erodin in response to ARC2. And then one of the surprises was then when she knocked out the serine threonine kinase, it had actually no effect on the response to ARC2. And so we think that the old model for how this signal transduction pathway works is incorrect, and that in fact AFSK has nothing at all to do with antibiotic production. And in fact, colleagues in the UK published a paper some time ago that showed that the actual role of AFSK is to regulate cell wall production. And it does so in a complicated manner, but it mostly involves phosphorylating the protein Div4A, which some of you might know as something that recruits peptidoglycan biosynthetic enzymes to the cell envelope. And so we think that that is the real role for this pathway now, and that there's presumably some other kinase that is responsible for phosphorylating AFSR, and we have a search on now to try and figure out what that kinase is. So that's where we are with that project. So hopefully that sort of illustrates the kind of thing that we learn. Start with a really simple idea. Can we turn on blue stuff with chemicals? And we found some. And then we've used that at once to discover new natural products, some of which have some promise, at least as to study. And they could have promise as drugs. Those are the 2 antifungals I talked about. And we have other compounds we've discovered through this endeavor. In addition, it's given us kind of a unique way to look into the cell. Chemical probes chemical elicitors like this are terrific tools for looking at perturbation of gene expression. The combination of a bioactive molecule and RNA-seq is incredibly powerful. And so we're sort of using this strategy on many different compounds that we're studying now. So, any questions about that before I move on to part 2? Okay. Okay, so chemical perturbation of spore formation. Why would we do this? Hopefully that will become clear. We've just published the first 2 papers on this project, and it's been the most unusual sort of set of surprises, and well, it's the origin of my title. We had one big surprise and one thing that was surprising because it was so unsurprising. So I'm going to tell you about 2 stories that have come out of the work on spore formation. In one case, we found an inhibitor of spore formation that turns out to hit some new antibiotic target. We don't know what it is, but we know that there's no other antibiotic in the world that does what it does. And the second, we have an inhibitor of spore formation that hits one of the best known antibiotic targets ever, and that's also exciting. The compound itself is very exciting. So let's go back here for a second. So the sporulation part of the life cycle of Streptomyces bacteria involves the erection of this white fuzzy layer of cells on the colony surface, and these are the aerial hyphae that grow up into the air. And they do a bunch of things that the substrate hyphae don't do. I should say that most of the secondary metabolites are made in the substrate hyphae, and the aerial hyphae are where the spores are made. And this happens through this developmentally regulated round of cell division and morphogenesis that converts these compartments into individual cells that can then be released. And in nature, you imagine them blowing away in the wind and moving the colony to another place. So why would we want to chemically inhibit this? So there's a couple of reasons. First of all, it's just an interesting way to find out how this mechanism works. The other thing about this is that one thing we thought at the beginning was that we might find molecules that interfere with the cell division apparatus. And those of you who know about antibiotics know that virtually all of the antibiotics that we have target the cell wall, RNA polymerase, DNA gyrase, the folate pathway, and the ribosome. And there's a few other outliers, so triglycerin is fatty acid synthesis, but aside from that, it's really those 5 processes, and that's pretty much it. And the cell division apparatus is considered by many people to be quite a good drug target, or things around the cell division apparatus. So that was our initial motivation. We did actually find one cell division inhibitor, which I'm not going to talk about. I think it's probably not that useful a molecule. But we've learned a whole bunch of other interesting things about how cell division takes place and is regulated inside these cells. So first of all, let me show you what the data looks like. So you recall me mentioning Streptomyces venezuelae, which is the species that makes a green pigment, deposits a green pigment in its spore wall once the spores mature. So you can see that this is a lawn of Streptomyces venezuelae, and it has a really beautiful green pigmentation all across it. And then there are sterile filter discs added with different molecules in them. So there's a couple of solvent controls. And then there's 2 different compounds below that are inhibitors of sporulation that also came out of Aaron Craney's screen. I should actually backtrack a little bit and say the molecules that Aaron was most interested in were the molecules that perturb secondary metabolism. But she also found molecules that accelerate sporulation, molecules that inhibit sporulation, all kinds of different molecules of interest. And all of these molecules have an interesting sort of 2 MIC phenomenon in common. So the MIC, for anyone who doesn't know, is the minimum inhibitory concentration of an antibiotic. So one way to describe the potency of an antibiotic is by the lowest concentration that will kill the cells you're interested in killing. So the lower the MIC, the more potent the antibiotic. And so what I mean when I say there's a 2 MIC phenomenon going on here, it's this. With all these molecules, so this again is the filter disc on a lawn of Streptomyces venezuelae. There's this one region of cleared growth where there's no cells at all. And that defines an MIC for growth. So these molecules all have some antimicrobial activity. And then there's a second MIC that's defined by the edge of that white ring. So the white ring is an area where sporulation has been prevented. And we know it's been prevented because you don't have the green pigment. When the cells finish sporulating, they make the green stuff. And in fact, you can look at this microscopically, and you can see that whereas normally what you'd have at this stage is just great long chains they look like linked sausages of little, clearly unicellular spores linked together. In this white region, what you see is these long filaments, long aerial filaments with no successful septation at all. Okay? So I'm going to tell you about 2 molecules that came out of this screen. The first is called Min1, and the second is called EN7. So both Min1 and EN7 have this property. They have if you had enough of them, they're antimicrobial to Streptomyces, and at lower concentrations, they block the sporulation process. So let's start with Min1. One of the really gratifying things about this was that almost all of the compounds that we found that were inhibitors of sporulation in Streptomyces, if we tested them against bacteria that were pathogens, in particular Staphylococcus aureus and Enterococcus faecalis or faecium or any of those guys, they were pretty potent antibiotics. Min1 was one of the weaker ones. It has an MIC of about 30 micromolar, which is not astoundingly high. But the exciting thing about it was that it was active against clinical isolates of MRSA in the case of Staph aureus and VRE in the case of the Enterococcus species. And that's important. Those are both clinical superbugs that are a serious problem and growing problem worldwide. So that attracted our attention. That's how we decided to work on this particular molecule. And we started just sort of doing things with it to see what it would do to these cells. And the most interesting effects that we had were actually in Bacillus subtilis. So I'll show you some data here. Bacillus subtilis, it depends a little how you grow it, but usually if you grow it, it grows in chains. They're single cells, so if you can see that you have a whitely stained, intensely stained area at the end of each cell, those are each individual cells, and eventually those will pinch off and you'll have individual bacillus cells. So it's not like Streptomyces. It's not a filamentous cell organism, but it does grow in chains, and then they have to break apart. So if you compare these two pictures, so on the left you're looking at Bacillus subtilis growing in a control culture, and in the right you're looking at Bacillus subtilis growing in the presence of a low concentration of Min1, just below the Min1 MIC. And I don't know if you can see it, but reproducibly, the Min1 treated cells are shorter, which is fascinating. There's there's a little bit known, but one of the really curious mechanisms and unfinished mechanisms in microbiology is what it is that makes all cells be the same size. Min1 has this weird property. I don't know if there's some antibiotics that interfere with fatty acid synthesis that do something like this. But for the most part, most antibiotics do not. When they kill a cell, the cell stays the same size and then it blows up. With this case, the cells shrink. And they shrink by a defined amount. So they go from at the concentration, our working concentration, which I think is usually 9 or 10 micromolar, they go from 5 microns long to being 3.5 microns long. So when we first started working on this, the only clear regulator of cell size that we knew of in Bacillus was the UGTP pathway. It's a really unusual pathway. UGTP is a mechanism that takes glucose and converts it. So it's a metabolic pathway, and it converts glucose into various glycerol-related sugars that the cell uses for various purposes. And for some reason, one enzyme in that pathway, UGTP, directly binds to the protein FTSZ and regulates cell division. It's a direct regulator of cell division. So dual-purpose metabolic enzyme, regulator of cell division. So we worked with collaborators at Washington University, and we came to, and we've done a whole bunch of experiments to basically, mostly what we succeeded in doing is figure out what Min1 does not do. On the left, you're just looking at a graphical representation of what I just showed you, which is that as you titrate in Min1, the cells get shorter. That effect plateaus at about 9 micromolar, where the cells are stuck at around 3.5 microns long, whereas they're normally about 5 microns long. So we worked with Petrolevin's lab, and they have mutants in the entire UGTP pathway. And so if you look at the black bars on the right, you can see that when you make mutations in UGTP, GTAB, and PCGA, or PGCA, the cells are shorter by a defined amount. What we found is that if you add Min1 to those cells, they get shorter still. And in fact, you can do almost the same titration, and you take the cells from the length they are in the mutant background down to 3.5 microns. So there's a whole bunch of other data in that, so it's a very complicated story, and I think I'll leave most of that out. It's published now if anyone's interested. And our current working hypothesis is that Min1 does something that affects a value called delta. So at the moment it's basically a mathematical model of the regulation of cell size. And the model is a very simple one. The idea is that if the cell grows by the same length, or it works length and volume, the same every generation, if it grows by the same amount every generation and then divides in the middle, then all the cells will be the same size always. So if you think that through, that's clearly the case. And in fact, there's nutritional tricks you can do to perturb cell size, make cells bigger or smaller. And if you imagine cells starting that way and then you return them to growth in LB, which is what we're describing here, then if they go back to growing by a delta of 5 every generation, then dividing in the middle, they will slowly return to the size that they're supposed to be. And so the evidence from the work that we've done so far is that somehow Min1 acts on delta, and it changes it from a value of 5 to a value of 3.5. And it somehow does it through an interaction with the cell wall. And that's basically what we know at this point. That's exciting to me because there's very few chemical tools to dissect cell size in bacteria. And so one of the things we want to do next is find out what is it that min1 actually binds and what is the consequence of that. Okay. So the last thing I want to tell you about is another sporulation inhibitor that called EN7, which, as you're going to see, interacts with one of the best known antibiotic targets that we have. So this is the structure of EN7. Of all the molecules that came out of this screen, this is the most potent antibiotic that we have so far. This is a primary screening hit, so this is just, these compound libraries are made out of molecules that have been produced by chemists. They weren't made for a purpose except to be soluble and stable, and they weren't designed to be antibiotics. And so the fact that this one is so potent is remarkable. So these are cultures of Staphylococcus aureus, and this is a titration of EN7 going from 0.04 micromolar up to 20 micromolar. And you can see that it defines a pretty clear MIC at around 0.6 micromolar, so a sub-micromolar MIC on a primary screening hit is quite potent. So again, how does this molecule work? How do you get from this phenomenon to knowing what this molecule does? And so one of the tricks that we do is we have a collection of reporter strains we have various organisms, mostly in Bacillus subtilis, where we have stress responses tagged to the LAC-Z gene, so that if you activate the stress response with your molecule, the colonies turn blue. And so often we'll just take a molecule out of a screen and just add it to all these reporters and see what happens. And so we did that with EN7, and we found the following. So two of the reporters that we use a lot, YNEA and DIN-C, are reporters around DNA damage. So if you damage the chromosome, these genes are activated, and there's a whole response called the SOS response that then takes place. So if you look, so this is kanamycin is a negative control. Kanamycin does not damage the DNA. It binds to the ribosome, and you can see there's no activation of either reporter. Mitomycin C is a well-known DNA cross-linker, and you can see that it has a very robust activation of both reporters. And then it's faint, but if you look, EN7 induces both of them as well. So there's a little blue ring here and here. And so we thought, DNA damage. And then we went to the freezer, basically, and we pulled every DNA damaging compound that we had off the shelf and tested them for the ability to block sporulation in Streptomyces venezuelae. And so we tested bleomycin, doxorubicin, so those are both anti-cancer drugs, mitomycin C, novobiocin, which is the DNA gyrase inhibitor, and ciprofloxacin, which is a different class of DNA gyrase inhibitor. And all 5 of these molecules block sporulation in Streptomyces celicoli. So one of the, in all Streptomyces, in fact. So one of the discoveries we made here, actually let me show you some data, just keep myself honest here. So at the bottom, you're looking at a close-up of a disk diffusion assay with kanamycin. So you can see there's a zone of inhibited growth, and then you have almost green almost right up to the edge. It's a little bit white right here, but that's just because the cells are growing slower. In contrast, there's almost no antimicrobial effect of ciprofloxacin on Streptomyces venezuelae at all, but there's a long zone of white colony surface where sporulation is not taking place. So it turns out that sporulation is exquisitely sensitive to DNA damage in the Streptomyces. And actually, this kind of makes sense. I told you that sporulation is basically just developmentally regulated cell division with a few other bells and whistles attached to it, the morphogenesis of the spore, if you will. In E. coli and Bacillus subtilis, one of the first things that happen when you damage the chromosome is that the cell turns off the cell division apparatus. And so you get a phenomenon where those cells become filamentous all of a sudden. This is, in fact, true of all organisms. Like human cells, for example. If you damage the chromosome, you get a G1 arrest. The cell won't go through its normal cell cycle. So it's just one of these weird things where Streptomyces has taken a fundamental mechanism, in this case it's cell division in the SOS response, and reorganized it in time and space so that it's mostly about the sporulation response. The vegetative hyphae don't care about DNA damage at all. Many of those cells die during the normal course of the life cycle. Okay, so how is EN7 damaging the chromosome? What we did was we took an approach that we often do with these things, and we isolated resistant mutants in Staphylococcus aureus. And you're looking at titrations of EN7 against the wild type, which is in blue. So you can see that potent antimicrobial effect below 1 micromolar. And then we have 5 mutants shown here, all of which are resistant up to at least 10 micromolar. It actually goes up to about 20 micromolar. In fact, we sequenced a total of 9 such mutants, and all of them fell in either one or the other subunit of DNA gyrase. So those of you who are familiar with DNA gyrase, it's the enzyme that is responsible for maintaining the negatively supercoiled state of the chromosome. It unwinds the DNA in front of advancing RNA polymerase and DNA polymerase. And if you don't have DNA gyrase, what happens is you get this overwound chromosome, and eventually the chromosome gets damaged. So all 9 mutants had one of 4 mutations. And it was intriguing that we had mutations in both subunits. That's unusual. Most gyrase inhibitors, if you get ciprofloxacin resistance, it's in one subunit. If you get novobiocin resistance, it's in the other subunit. I don't actually remember which is which at this point, but resistance in both subunits is rare. So there's the gyrase. I won't go through that. But I'll just show you where these mutations map on the structure. Let's see. So this is the Gyrase subunit here, and this is the other Gyrase subunit in gray. Whoops. And then this is Gyr-B up here, and then if you rotate this 90 degrees, this is the other Gyr-B here. It's behind on this side. And so if you look, you can see there's one of our EN7 resistant mutants, and this is buried in there as a ciprofloxacin resistant mutant, or if you look in this projection, you can see it a little better. There's ciprofloxacin resistant, there's EN7 resistance. And you can zoom right in. That's ciprofloxacin resistance, EN7 resistance. And so the resistance determinants, when you get resistant mutants in this gyrase, they're in different places. And in fact, the EN7 is active against all of the ciprofloxacin resistant mutants that we've been able to pull. We went to a database of clinical isolates that are sequenced and that have mutations that are resistant to ciprofloxacin, and in fact many other antibiotics. And we found that EN7 was able to outright kill 70% of antibiotic-resistant clinical isolates, regardless of the mode of resistance. So we're quite excited about this. And then one last detail about this is we reconstituted a gyrase reaction in vitro. So you can take an unsupercoiled DNA, a circular DNA, interact with gyrase, and then because it becomes all supercoiled, it runs more quickly. And then as we titrate in EN7, you can see that this reaction is inhibited. So in fact, it inhibits DNA gyrase quite efficiently inside the cell, in vitro. In fact, it's almost 3 orders of magnitude more potent in a test tube than it is against cells. It's a really rip-snorting gyrase inhibitor. So that's all I want to tell you about today. I just want to summarize, to sort of summarize what we're trying to do is the idea is to use molecules to understand about how cells work, to use molecules to find antibiotics, either natural products or in fact the synthetic compounds themselves. And it leads us on some really interesting journeys where we find ourselves studying DNA gyrase with no preparation or warning at all. I've told you about a poorly understood mechanism. We have now a very specific inhibitor of a quite poorly understood mechanism, cell size. A novel inhibitor of a well-known, a really well-validated antibiotic target. And we're currently trying to optimize EN7 and Min1 to make them more potent and more bioavailable and all that kind of thing. And one of the goals of that is to be able to do animal trials with these compounds at some point in the future. So I'll leave you with that. I'll just acknowledge the team. So the students who did most, actually I'll just go right to the picture. So we have collaborators at Washington University. Oh, I left Petra off. Petra Levin and Stefan Vadia were very helpful. Tony Maxwell and Susan Schlimpert at the John Innes Center have also helped us in this work. This is the lab. The work that I told you about, Aaron Craney is not in this picture, but EN7 and Min1 were done by this guy, Scott McCauley, and Vanessa Yoon did all the AFSRS work. And so here we are looking all very serious, and then they emailed me this picture yesterday when I was in the airport to show that they were having a very nice time with their Halloween. And so I'll leave you with that. So thank you very much for your attention.
Thank you. Okay, questions for Dr. Nodwell?
Justin, in the first half of your talk there, you were talking about activating these cryptic gene clusters, your chemical stream, and it looked like the two molecules that you sort of activated from these cryptic clusters both appear to be polypeptides. Do you think that's just a happy coincidence, or do you think you're able to sort of selectively activate that?
So I don't know that we have enough data to really answer that unequivocally. I can tell you, so for example, another one of the molecules that we activate is CDA, a calcium-dependent antibiotic, or daptomycin basically, which is non-ribosomal peptide. Okay, so another thing I didn't tell you about, just because it's a big, hairy genetic story, is that ARC2, its direct target is Fab-I. So it's actually, if there's some chemists in the room might have noticed, it's chemically similar to triclosan. Triclosan is a covalent inhibitor of Fab-I. ARC2 is a non-covalent inhibitor of Fab-I. So at first what we thought it was doing was, so we're confused about this. This is another reason why I didn't tell you the whole story. At first what we thought it was doing was liberating CoA-associated carbon to be shunted into the polyketide pathways. And in fact, there is an acetyl-CoA needed to make daptomycin, so that fits. There's a little chain of carbon attached to it, along with all the peptide stuff. But that turns out to be wrong. We went on, we actually measured fatty acids being produced by cells in the presence and minus of ARC2. And in fact, what happens is that fatty acid levels stay the same. Fab-I is an enolase, so it removes double bonds every round of Claisen condensation. And what happens is that ARC2 actually drives up the amount of unsaturated fatty acid. So our best guess at the moment is that what ARC2 is doing is some kind of stress response. Maybe these molecules are being incorporated into the membrane somehow in the form of unsaturated lipids. The cell doesn't like that, and it activates a stress response that acts through the conduit of AFSR and AFSS. But that's officially what hand waving looks like. So.
I'm curious about what happens to EN7 when you test against the biosyn-resistant?
Full activity. It's active against novobiosyn resistant and Gyrase and ciprofloxacin resistant, at least in staph. We haven't gone to any others. And this is why I was interested in your molecules. It does absolutely nothing against gram-negatives. This is the bane of my existence. We can get antibiotics against gram-positives, and it's relatively, I see people nodding, it's not so hard. But to get antibiotics against gram-negatives is Herculean. In all the time we've been looking for antibiotics, we've found one molecule that was potent against gram-negatives. It was amazing, and it was a screen against Burkholderia. The problem was that the molecule had a half-life at room temperature of about 6 hours, and then it fell apart. So that was no use. So the answer is that the ciprofloxacin resistance and novobiosyn resistance have no effect on EN7. If you look closely, EN7 may be related to some of the NBTI compounds that came out of Merck a couple of years ago, which, as far as I know, they're not working on anymore. But we don't know if that's meaningful. But the next step is definitely to get a structure of Gyrase with EN7 bound to it, and we've got a collaboration set up to do that. Don't be shy, people.
During sporulation, so when you close DNA damage, does that block replication and segregation because of a physical block, or is it a checkpoint-mediated process? And if it's the latter, and you abrogate the checkpoint, does that restore cross-activation?
That's an awesome question. So Jan Falguera, this guy here, never send your supervisor a silly photograph. So he spent the summer, he's been working on what the actual mechanism is. And so we have RNA-seq data. And what we find is, some things are predictable. If you treat Streptomyces with EN7, you see a small drop in most of the cell division genes. So twofold, threefold. But there's one gene called SSGB, which is only found in the Streptomyces, as far as I know, it's thought to be, we don't really know what it is, but it binds to FDS-Z. It may be one of these Z stabilizing proteins. That one drops like a stone, like a 12-fold drop in 2 division cycles. So it's got to be active. It's got to be some proteolysis. The RNA vanishes. There's got to be some, like the cell says, no more of this, and gets rid of it. And so what Jan has done is he's created a strain in which he can induce SSGB independently of its normal promoter. So anyway, it doesn't matter what the promoter is. We can turn SSGB on and ask what happens if you tell the cells, you don't want SSGB, well, you've got it. And now we're going to damage your DNA, and let's see what happens. And the answer is they make spores. They go on, they complete the life cycle, they make green pigmented spores. So I think it's a checkpoint. I mean, the bar is high to say something is a checkpoint. So this is the first time I've said it out loud. Yeah, and the next thing we want to test is we want to collect up a bunch of those spores and test them for evidence that they have DNA damage, because the logical assumption is that the cell does this for a reason. You don't want to send a bunch of babies off into the world with wrecked up chromosomes. And presumably, when you circumvent the DNA damage response in this way, that's exactly what you do. So we're going to screen for oxytrophies, whatever, anything we can look for. So that's a terrific question.
Regarding the cell length assay, there are quite a few bacteria that are pleomorphic, especially in unfavorable conditions.
Yep.
So how confident are you that it's actually interacting with cellular division rather than, say, influencing fatty acid production so the cells aren't so long?
So that's a good question. We know, what do we know? So it actually interacts with the cell wall. I don't think it actually directly interacts with cell division. It does something in the cell wall, is all we know. And we know that because it activates, you remember those reporter things I mentioned. I had the data in initially, and then it was just too confusing. There's a whole bunch of different mechanisms that respond to damage to the cell wall. There's a gene called YWAC that responds to any kind of damage to the cell wall. Sigma W responds only to damage to the peptidoglycan. LEA-I responds to lipid 2, blah, blah, blah. And we see a very specific kind of fingerprint that this molecule has that it activates some of these stress responses. Not all, but some. So it's doing something in the cell wall that is somehow relevant to the setting of delta. Now, have we looked at other growth conditions? We have. And what we find is that if you really starve cells, so if you grow cells in a defined medium like an M9, they're smaller. I mean, we didn't discover that. Petrolevin discovered that. Oops, sorry. And so the smaller the cells get, the less effect that Min1 has. So for example, if you grow in just a defined minimal medium, the cells are already at about 3.5 microns or maybe even less. Min1 has no effect under those conditions. It mostly has an effect when the cells are fed. So I rather doubt that it has anything to do with nutrition, just because they're sitting there with a giant smorgasbord of LB, right? They have all the amino acids, they have and the carbon source and all the rest of it. They don't have to make anything much. So.
Just wondering if, so EN7, you said it has no activity against gram-negatives, right?
If we use one of the strains that has a compromised outer membrane, then so that's why I think your molecules might be of interest. The answer, it's very potent, yeah.
Let's say a combination of cyclophosphates, what kind of interaction?
We haven't done that, actually. Yeah, I would think I'd be more interested in something that compromises the wall, like even a beta-lactam or something like that. Because I assume the problem is the outer membrane or efflux or something like that. Although, yeah, we don't know. I haven't done the experiment, is the truth of the matter.
I'm not sure if I saw this right, but when you showed the Min1 molecule, I thought, didn't R2 also have this little group of 3 fluorines?
Oh, yeah, so that's a good eye. Yeah, so that's true. And that's, So most of the molecules that go into small molecule libraries come from combinatorial chemistry. And so it's basically, you start with a scaffold that has a bunch of reactive centers, you block most of them and you modify one of them, then you deblock another and you modify that, and you sort of build up complexity sort of in I don't know what anyway, you just build up complexity. The fluorine is a blocking agent. So many small molecules in compounds in synthetic compound libraries have fluorines all over them. And so I don't think those fluorines are relevant to function, although we haven't tested that. Actually, in the case of Min1, we have, because we did a pretty big, what's called a structure-activity relationship, where we tested a whole bunch of analogs, and so we have a pretty good idea about what is important on Min1 and what is not. And the fluorines are just kind of decoration.
One last question, yeah?
In that picture before dissipation, you draw this long tail, right? So what's this tail is? I'm just wondering, is it a single cell so long?
Yep, it's a single cell. It's one cell. Yeah, on the order of 50 chromosomes. So the substrate hyphae, they typically grow to have, I don't know, people argue about exactly how many, but certainly 20, 30, 40 chromosomes per compartment, and then they'll lay down one of these cross walls, which is just and actually how those cross walls are made is not known, although people are studying it. And it's analogous to a syncytium in a fruit fly embryo. The aerial hyphae are longer, so, and one of the differences between aerial hyphae and substrate hyphae is that substrate hyphae branch, aerial hyphae do not, so they're just a single long cell. And then I debated showing them. Jan brought back videos of these taken in a microfluidic device. It's fascinating to watch because you can trap a cell in a microfluidic device that is expressing a GFP FTSZ fusion. And you can watch, it will sit there, and then it will light up with septa as all the FTSZs, as this gene is turned on, and you make the Z ring at the sites of septation, and it's almost perfectly synchronous. It's really quite marvelous. So that's one cell. It's an unusual bacterium. I mean, all bacteria are fascinating, but I'm partial to this one.
Great. Okay. Well, if you join me in thanking Dr. Nodwell for an incredibly interesting talk, and I'm sure